So our first brave speaker is going to be Ruth Arnold. Oh, and speakers, stay in front of the podium where they can't hear you or see you online.
Okay, fantastic. Well, I hope everyone can see and hear me okay. I'll try not to move around too much. As Dave mentioned, my name is Ruth. So I'm a brand new master's student here at the Institute of the Oceans and Fisheries. I'm in with all the fantastic team at Project Seahorse. And I'm going to be telling you a little bit today about me and also about my research, which is on population assessments for the 3 seahorse species of the Yucatan Peninsula in Mexico, and maybe why you should care about that as well. Okay, so a little bit about me. My background is actually rooted in trying to understand the illegal wildlife trade. So I was doing my research out of Oxford on Saiga antelope. And I've put a few of my outputs on the screen. If you're curious, come and ask me about that in the break. But I worked alongside CITES and Traffic and then presented my work at the COP14 in Samarkand this year, CMS. So I made a little bit of a transition because I fell in love with the marine world. And now, thanks to the Oxford to Canada scholarship, I'm here to now focus my research interests on marine conservation and how we can do that also with local engagement and community work. If you haven't seen me around department very much, that's because, as you can see pictured, I've been doing lots of diving preparation. So the darker image is BC Waters, where I've been doing my Caus one. And then on the left was where I really fell in love with diving, which was in Indonesia. So yeah, super grateful to be here. So why seahorses? Now, I've stolen this image from the Project Seahorse website. And to start off with, for all of the diehard seahorse fans, I have to mention they are both beautiful and interesting creatures. They are also populations at risk worldwide, but they're also very attractive species for establishing marine protected areas, limiting fisheries, and regulating trade. So you can see how we move from that organism level all the way up to potential worldwide and nationwide policy. So we like seahorses. So why Yucatan? So Yucatan is of particular interest because of its position as a growing area of conservation concern, and also for their presented interest in seahorses and the populations found there. So here I've provided a map on the upper right-hand corner from iNaturalist, where we can see these clusters of seahorse species and a lot of work being done by UNAM, which is the university in Yucatan in Merida. Now, if we dive a little bit into these records, we find the lined seahorse, which is Hippocampus erectus. Now, the reason that this research is important in this area is for a number of reasons. The first of which is that Hippocampus erectus is both vulnerable and decreasing, according to IUCN. So it is, of course, of conservation concern. We're also completely lacking data. Not completely, but we're lacking a lot of monitoring data for Hippocampus erectus, radii, and zosterae. And all of this information is really important for us to understand how to implement CITES and national plans of action. And this will also involve trying to understand the regional risks faced by these species in the Yucatan. I should also mention it's probably one of the first studies to be doing this kind of assessment in the Yucatan Peninsula, so it's very exciting. We don't love the word baseline because it doesn't imply that there's going to be immediate policy benefit always, but we're hoping that this will be able to be implemented really quickly after the research has been completed. So the objectives: to gather some information on the known likely location and threats to the 3 target seahorse species, focus fieldwork to the potential sites, and then conduct in-water analyses of species populations and environmental variables. And at the heart of this, we really want to co-create the science with our colleagues from UNAM, marine technical schools across the Yucatan, and also with local fishers. How are we going to do that? Well, as you can see, this is very broad, and deliberately so because we're very short on time. But we're going to be running some stakeholder interviews, primarily focused on fishers, some local divers, and academics. We're going to use this information. This information will also, by the way, include trying to understand potentially some of the impacts of trade or impacts of local risks, as well as where the seahorses actually are. So it's almost like building into that form of a risk assessment too. Then mapping those potential dive sites, using that interview and iNaturalist information, and also trying to cover a good geographical distribution within the Yucatan. And then completing underwater visual surveys of the seahorse populations. And this will involve some photography, GPS logging, and also mapping some environmental variables. And if you're curious to know a bit more detail about what that could involve, do come and speak to me at the end. But we're very excited, as we're hoping then that all of this research can come together to help to formulate plans for national plans of action, like, as I mentioned before, better policies and also marine protected area establishment, etc. And that might evolve into doing some modeling of these potential management scenarios too. That all being said, I welcome lots of thoughts. As I said, come and find me. It's still very much in the shaping stages. So thank you for listening. I hope you enjoyed.
Yeah. Now we'll see how difficult it is to switch presentations.
Oh, see, it stops sharing. I don't see that. Okay, what's the
More.
This one?
Window.
This one?
Yeah. Oh, it's Deniz now? Oh, sorry. Just
Go where?
no. I'll go away.
No, go where, I'm saying.
Yeah. Sorry.
You're good. Okay.
Yeah, it's just trying to get it online.
I'm gonna try not unsharing next time, but we'll see if that works. Alrighty, I don't have my presenter's notes, so hopefully it's gonna be okay. Hi everyone, my name is Deniz. I'm a first-year master's student in the Pelagic Ecosystems Laboratory. I started in May. And yeah, so for my project, I'm going to be looking at the dynamics and drivers of the nearshore zooplankton community in Malaspina Strait, which is up on the left here. And I'll be using Burrard Inlet as a comparison to look at a more urbanized site. So why zooplankton? Zooplankton play a central role in marine ecosystems by transferring energy from phytoplankton to higher trophic levels. And when you see anomalies in zooplankton communities, that impacts those higher trophic levels. And there's a growing body of evidence that shows that when juvenile salmon encounter lipid-rich and super fatty zooplankton while they migrate, they grow faster, can store more energy, and have better returns. They also are great indicators of environmental health. They have a short turnaround time and are sensitive to ocean conditions. And so by looking at changes in the zooplankton community, we can get a snapshot as to the health of that ecosystem. And zooplankton assemblages in the Strait of Georgia are changing. So broadly, these changes have been linked with large-scale climate indices like the Southern Oscillation Index. And local drivers have only been suggested to be weakly correlated with zooplankton change. But that could be because data is an issue. Most zooplankton studies done in the Strait of Georgia are done at coarse temporal resolution. And a lot of them focus on mid-strait and deep water, like sections of the strait. And this coarse temporal resolution can limit our ability to look at aspects of zooplankton dynamics that are critical to ecological studies and studies of fish diets, like peak biomass timing and the timing of occurrence of critical prey species. And the samples that have been collected in the nearshore. A lot of them are sitting on shelves, collecting dust. Because yeah, microscopy is all really hard, takes a lot of work. Before I started as a master's student, I was working as a tech in the pelagic ecosystems laboratory, and I started working like doing microscopy. And as I was like spending those hours on the microscope, I was like, there's got to be a better way to do this. Turns out there is. This is the ZooScan. So it's essentially just a very fancy printer scanner that can take high resolution images of a zooplankton sample that look a little bit like this. Yeah, so these are images that I took with the ZooScan. And then these images are then automatically identified using deep learning in relation to a reference image library set to get taxonomic groups. And while this method is, you get like a less specific taxonomic resolution than microscopy, you can get rapid and important information for studies that are like important for ecosystem health, such as peak biomass timing, organism size, biovolume. And yeah, so my objectives through my work, the first one is to develop protocols for the accessible and widespread use of semi-automated image processing technology in BC. Right now, there is no publicly available reference image library for the Strait of Georgia. So I have been already working to build up what will be eventually a publicly accessible reference image library that I'm hoping will be ready by the time I graduate. And I want to use these methods to assess seasonal and interannual zooplankton dynamics in Malaspina Strait and how they relate to oceanographic drivers, and assess spatial variation in nearshore zooplankton community metrics as they relate to oceanographic drivers, including metrics of human disturbance. So to do this, I'm going to be using Pacific Salmon Foundation's citizen science project that has been going on since 2015. And they collect a variety of oceanographic metrics throughout the Strait of Georgia in about like 80 different locations. 3 of these locations include zooplankton, and those are the ones up in Malaspina Strait, which is a critical migration corridor for juvenile salmon. And for the purposes of this project, all of these sites are being considered as nearshore, and they have varying depths from 30 meters down to 420 meters. So I've been able to get some preliminary results from the 2023 and 2024 Malaspina Strait samples. And despite having the highest density, the shallowest site has showed the lowest species richness and highest variability, while the 2 deeper sites were a lot more stable and similar to each other throughout the seasons. Yeah, so the low biovolume in winter could be due to less large overwintering species, and it's likely that the shallowest site is comprised of smaller species with higher turnover. Yeah, so I have a poster where I presented the results I just talked about and some more at Pisces like super recently. It's on the 3rd floor. If you want to learn more, go find it, come talk to me. And yeah, thank you. Where it unshares, it's
No.
not supposed to do that.
Oh, no. Cooler. Oh, dear. Oh.
Okay, hi everyone. My name is Stephanie. I'm also in the Pelagic Ecosystems Lab, where I'm going to be investigating urbanization-driven changes to ecosystem structure and function. And I don't know how to change slides. Okay. So a little bit about how I came to be in the IOF. I originally studied chemical engineering and I worked for a number of years in the charity sector as well as in industry and R&D. But at some point during that, I'd also started to live full-time aboard a sailboat. And I was fortunate enough to have the opportunity in 2019 to sail shorthanded clockwise around the coast of the UK. And it was during this experience that I started to really get an awareness of the myriad ways that we're impacting our marine environment. And that's what ultimately drove my decision to retrain in marine science. So for my master's, I looked at characterizing and quantifying threats associated with vessels and coastal marine life in an area known for heavy dense marine vessel traffic in Southwest England. And that leads me here, where I'm going to be looking at the impact of urbanization. So I'm interested in quantifying trophic pathways in a heavily urbanized nearshore pelagic food web and looking at key stabilizing processes, so trophic compensation and functional redundancy in those ecosystems, exploring if there are any key identifiers that are specific to an urbanized versus an unurbanized environment, and predicting what this might mean for the future of these ecosystems. So the majority of research that's looked at responses to ecosystem impacts from anthropogenic ecosystem changes from anthropogenic impacts has looked at climate change. So there's been studies that have shown an overall reduction in diversity without being dependent on a species' resilience or adaptive capacity to different environmental drivers. Also changes in food chain length with the number of trophic steps having an impact on energy transfer efficiency to higher trophic levels, and overall shifts in interaction strength. And that can be from things like different consumption rates and phenological mismatch. So the figure on the right there is from a mesocosm study that looked to replicate the predicted acidification and warming scenarios. And it showed significant changes in the trophic structure. So the blue represents increases in connections between predators and prey, and then the pink represents decreases. So why is it important that we specifically understand the impact of urbanization? Well, coastal cities are rapidly expanding. There's been more than a 26% population growth in just under 2 decades. And 50% of that increase has been in what's classified as ultra-coastal, so within 10 kilometers of the shoreline. That coastal urbanization is predicted to continue to increase, and with that comes increased land-sea interactions. So greater areas of impervious surfaces, larger volumes of wastewater and stormwater runoff, as well as habitat degradation. And these urban stresses can be broadly characterized into 3 main types: ocean sprawl, exploitation, and pollution. And each of those have the potential to impact the health of a marine ecosystem, as well as their cumulative effects. So the ways that I'm going to be assessing ecosystem function in these urbanized waters is firstly looking at the influence of terrestrial inputs on trophic dynamics. So if and to what extent these additional material fluxes are being taken up into the marine food web. Also, whether there are any distinct differences in the zooplankton assemblages in proximity to urbanization, with a focus on grouping by functional traits, and whether there are any responses to urban stresses in shaping food web structure. I'm hoping to use all of that to develop a model that can project how expanding urbanization may impact like the stability of marine ecosystems in the future, and potentially proposing a set of urban-specific indices for ongoing conservation and monitoring efforts. So my research is going to be focused on Metro Vancouver. The city is located in a particularly interesting area, being situated in a coastal temperate rainforest ecoregion that already has significant land-sea connection. And I'm going to be building towards the growing research in Pelagic Ecosystems Lab under the Urban Oceans Project, predominantly in Burrard Inlet. And the idea is that whilst Metro Vancouver is being used as a case study, this could be applied to other coastal cities around the world, developing our understanding of what the impacts that urbanization may have on these ecosystems. Okay, so thank you very much, and I'm happy to answer any questions.
Everyone's looking at this. Alex. Yep.
Okay.
Alex.
Hello. Yeah. So my name is Alex Schmill. I'm an MSc student at the OCF, and I'm working in William Chung's Changing Oceans Research Unit, as well as a Hackeye Institute research technician. So a little bit about me. My research interests include mapping biodiversity and species distributions. I'm also interested in understanding more about species movement and habitat use, and just general data science. And some of my hobbies include diving, surfing, and making shiny apps on the side. If you don't know what those are, come talk to me later. So a little bit of background about the project. In 2023, the government of Canada and the Mapakuk First Nation began plans to start assessing the feasibility of instating and establishing an NMCA, National Marine Conservation Area, in the south coast of Newfoundland. And as part of that assessment, they seek to understand what species might benefit from this protection. So I partnered with Mapakuk Guardians to essentially understand the total biodiversity of the area. And in doing so, we designated 9 sites that kind of move from an open ocean gradient up a fjord. And that little star is Con River, where the Mapakuk Guardians are based out of. The idea is that we were going to sample 2 different depths at each site and do that throughout the year. So in spring, summer, and winter, all using environmental DNA. It's a little bit of background about environmental DNA if you guys aren't exactly familiar. eDNA, environmental DNA, is any genetic material originating from microbes or shed by organisms that are collected from environmental samples, and that can be water, soil, or air. The workflow generally for marine biology in our case is that we are going to be sampling with Niskin bottles at those 2 depths, and then filtering out the water with really, really fine filters, about 0.22 micron, to let the water pass through and catch all that genetic material. That genetic material is then amplified and sequenced, and then we can identify everything from microbes to mammals. And while I don't think Jacques Cousteau was thinking about eDNA with his quote, it really resonates with me. The ocean is the soup of life, right? So eDNA, why is that the tool that we're going for? It's super, super sensitive, and it can detect trace amounts of DNA, which makes it perfect for detecting rare, cryptic, invasive, and migratory species. One sample can yield hundreds of species detections, and all without extracting or disturbing the ecosystem at all. It's easy to implement, it's very, very fast, and it's relatively expensive, so it makes it quite scalable for future biomonitoring. And this is just an example of the data you might get from an eDNA study. So the numbers across the top might represent individual samples or different sites. And you can see that it can give us information about the species that are present and their relative abundances. So the ocean, it's a very dynamic place, and the species that occupy the ocean are obviously reacting to that change. An issue with a lot of environmental DNA studies right now is that they're just taking one snapshot of biodiversity, and they're not taking into account the whole temporal aspect of the ocean. If we can start sampling throughout time, it can fill in some of those data gaps and give us some really, really interesting insights about what species are moving in and out of the area and to what degree and abundance they are. Another way of understanding species distributions is the species distribution model. However, these models are only as good as the data that you feed them. And sometimes for different species, there's not much data. So I see an opportunity here with eDNA occurrences filling that data gap, especially being able to sample really hard to access environments and detect really hard to detect species. We can help improve this potentially. There are some improvements in SDMs right now. Something called a dynamic species distribution model basically predicts habitat suitability over space and time. And eDNA can confirm this basically as a power couple. We can effectively predict where species are going to occur and then use eDNA to not only validate it, but give us a sense of the species occupancy in terms of a prediction value as well. So power couple indeed. And that's exactly what I aim to do with this research. I want to combine these 2 methodologies to answer 2 key questions. The first being, do eDNA detections align with the occurrence probabilities that DSDMs predict? And can we integrate eDNA with DSDMs to improve accuracy of the predicted biodiversity patterns that we're expecting? So the big picture for my project is effectively to improve the reliability of eDNA as a biomonitoring tool, as well as inform the marine spatial planning in this area to hopefully establish this NMCA. Another byproduct, basically my favorite part, is that we're also getting to train the Mapakuk First Nation to take data sovereignty and sample their own waters and analyze it themselves to keep their data within the community. Yeah, that's about it. Thanks.
No. Yep. Good there. I don't know if it's sharing. Do you want to check the sharing? It's sharing. Yep.
All right. We're getting better. Who's getting?
Hello, my name is Rhys. I am a master's student here at IOF. I'm in the marine mammal lab, and Dave is my just said stupid advisor. That was a Freudian slip. Supervisor. Yeah, so I'm just gonna do like a little brief intro into myself and what I'm doing here. Yeah, so for the past few years, I was working in ecotourism. I worked in South Africa for a bit, Tofino for a while. And yeah, it was kind of a time where I developed an appreciation for charismatic megafauna. But yeah, then I kind of got tired of ecotourism after a while. This image right here, this really resonated with the marine mammal people, but this is the average look of a guest aboard a whale watching boat. It's very hard to please everyone. Julia knows. No. And yeah, around the same time, I was kind of losing interest in ecotourism. I was becoming interested in plastic pollution and how it might affect whales. Wasn't sure what to put here, but AI helped. Oh my God. Scary, huh? Yeah, so my general question is, are humpback whales eating plastic? And if so, how much? And why is it important? Well, it's really unknown whether plastic ingestion is a significant risk to humpbacks in BC. It's not currently listed on the COSUX latest report. Heat waves, vessel strikes, entanglements, and underwater noise are the big threats, but plastic pollution is nowhere to be seen. And I assume that's just a lack of data, not so much a lack of threat. And then also this research should provide baselines that can be compared in the future. If it's getting worse, better, you know. And the theoretical approach, so whales may be ingesting plastics directly from the water when they're feeding. They take big gulps when they're engulfing swarms of prey, or those plastics might come from the prey themselves who have ingested plastic, so trophic transfer. And with my research, I'm going to model daily microplastic ingestion using the concentration of microplastics in whale feces and in the seawater. With the feces, I can do this conversion. I won't get into it now, but you convert the concentration of microplastics in the feces to what you would see in the prey and their, yeah, use trace elements. It's a whole thing. You can ask me later if you're interested. Yeah, so the field work, I should say too, the field work, we've already done this all. This was done in the summer and it was good. Our objectives were to collect whale poo and to collect subsurface seawater samples. Yeah, with the whale poo, it was really good. We got 20 fecal samples over about 3 months, which is really good. It's pretty hard to get whale poo. And we had really strict contamination controls as well to prevent plastics from affecting our samples. And yeah, everyone wants to see what whale poo looks like, so there you go. And yeah, so the seawater samples as well, we collected them with a device called the Ascension, and you can deploy it at whatever depth you want. We chose depths to kind of correspond to the depths we know whales feed at to get an idea of what they're exposed to. Yeah, it was a great success. Yeah. And so right now, currently, I'm starting lab work. We have all the samples. I'm doing lab work at PSEC, the van or the lab in West Van. Yeah, this is I don't really pay too much attention to this. The methods with isolating plastics in any biological sample, in this case species, you'd use a chemical that dissolves the poo and just leaves plastics behind, and then you can count and quantify it that way. Oh, here's some clips of whale. Pretty nice, bubbling away. There's some more there. Oh yes, I have a bit of a cautionary tale here for everybody. So there's two chemicals commonly used to dissolve samples to isolate microplastics. One is hydrogen peroxide and the other is sodium iodide. Normally these are used in isolation. I figured put them together, it'd be quicker and better. But it turns out if you mix hydrogen peroxide and any iodide salt, it creates a thing called elephant toothpaste, which they do in like high school chemistry classes. If you don't know what it looks like this. I'm not gonna use it. Everything's fine. Oh, yeah, I don't know how that got in there. Really embarrassing. Yeah, this research, it's funded by the Clackwell Biosphere Trust, Ocean Legacy Foundation. It's a organization I work with, and DFO. DFO's been, you know, it couldn't have happened without DFO. And this slide was for the Mammal Symposium. I don't think we have time for questions, so I'll end it there. Thanks.
Oh. Sorry, no notes.
I might be able to get you notes. Hold on. I think now it's.
Oh,
you get-
let me just check. I think so. Just for you. Yep.
Hi, my name is Julia. I'm one of many Julias here at the IOF. So to avoid confusion, you can call me Julia Mayer or German Julia. I've been around here for like 3 years now, so some of you might recognize me. So I was born and raised in the southwest of Germany, a pretty landlocked part of Europe. After I graduated from high school, I moved to Scotland to do an undergrad degree in geography and environmental science, and that was the first time I lived anywhere near an ocean. So after regular cold plunges and climbing up sea cliffs, I eventually realized that the ocean was more than just a backdrop for me. To learn more about it, I ended up doing a bunch of extracurricular courses at the University of Utrecht in the Netherlands and the Bermuda Institute of Ocean Sciences. Yeah, and then after I graduated from the University of Dundee with my Bachelor of Science, just a few months after Brexit, I decided it's time to move on and look for master degrees in other countries. So in September 2021, I moved to Vancouver to do a master's degree in geography here at UBC with Simon Donner to study the impact of the El Niño Southern Oscillation on wave variability in the Republic of Kiribati and how that will change with climate change, so with sea level rise and coral reef degradation. During most of my degree, I was based here at the IOF because I felt more connected to the people here and our joint passion for the ocean. I defended my master thesis exactly today, a year ago. In January 2024, I started a PhD here in Oceans and Fisheries under William Chang and Mesira Costa from the University of Victoria. We established a MyTax partnership with OceanWise, an NGO here in town. And OceanWise had just started their deforestation program. Deforestation refers to the restoration and management of kelp forests. And the aim of this partnership is to find climate refugia along the BC coastline to see where these kelp forests could thrive in the future under climate change. Kelp act as foundational species here in BC, with many marine species relying on their abundance and density for feeding and protection. It's also an essential element of food security and ceremonial practices to many Indigenous communities, with harvest practices deeply rooted into traditional knowledge systems. Gradual ocean warming and marine heat waves are increasingly impacting these kelp forests. So seaweed aquaculture and kelp restoration are often considered low regret strategies to enhance biodiversity, food production, fishery support, and greater socio-ecological resilience. However, marine restoration faces significant challenges, including grazing pressures and large-scale habitat loss due to increasing extreme events. Using an ensemble of species distribution models, encompassing statistical, machine learning, and rule-based models, my project will alleviate some of these challenges by determining which sites and species are particularly suitable to do these deforestation projects, focusing on future climate change impacts. This will guide OceanWise in their selection process of deforestation sites and species to allow BC communities to experience the long-lasting benefits of kelp forests. Thank you.
Okay. Last but not least. Looks good online.
Okay, so hi everyone. My name is Antoine Mesple, and I'll tell a story about the French trying to project the exposure of marine heatwave hazard for exploited marine species. So, like I said, my name is Antoine Mesple. I'm 21 years old. I'm a master's student in the Changing Ocean Research Unit for 5 months. I'm already halfway, so only 2 months left. And I discovered that I love to dive last year. And this is me. It could be anyone. It could be anyone, but yeah. So how did I get here? So this is little Antoine. He wanted to be a doctor to cure people and had thalassophobia too. So definitely not the profile for a marine researcher. But I had this really inspiring teacher and in high school, and he sparked my interest into going in that direction. So I chose the school Institut Agro Rennes in the fisheries specialization, and then I got here in the core units. So my research is about projecting exposure of marine heatwave hazard for exploited marine species in the Pacific Ocean. And for a bit of context, so a marine heatwave is a temporary event, an extreme event characterized by an uncertain intensity and duration, and can be divided in 4 categories, from the lowest tier, so category 1, to the highest tier, category 4. And marine heatwaves actually have a different impact regarding on the biome they occur in. So I had to, for the study, I have to completely separate the biome in my analysis. So here is a map of the biome I use, so polar, temperate, tropical, and the upwelling biomes. So for my research, I used a database of the prediction between 1971 and 2020 of the abundance of some species, but here I only took two of them, so the Pacific halibuts and the Pacific bluefin tuna. So since those species are not present in the same biome, as you can see with the percentage of abundance, I had to separate the graph for see the vulnerability to heat waves in the future. So I'm using IPSL 585 model, Earth model for now to project the vulnerability of the different categories of marine heat waves across time. And as you can see, it's pretty different for the both species. So meanwhile the Pacific halibuts will be more vulnerable to category 2. For the tuna, is actually the 4 category he will be vulnerable. So basically 25%, yeah, more or less of the whole category. So here So here the bluefin tuna will be more vulnerable in general. So yeah. So the goal and future direction, the goal is to highlight the most vulnerable species to watch for in the future. So the hypothesis is like pelagic species will be more vulnerable than benthic species, and to try to see if this is okay. I'll continue the analysis with more species and expand the study to all the oceans in the planets, and compare the different scenarios too. So IPSL 126 and the GFDR. Thank you for your attention, or like we say in French, merci de votre attention.